Hi, everyone. How are you all doing? Thank you all for attending. So this is the fourth session in our series, our career series for individuals that are interested in a field in or a career in clinical genomics. So this week we have a panelist of individuals who are in various areas, but they're all focused on research development and other opportunities. We do have the recordings available from our other previous three sessions. So the first one, we had a panel of genetic counselors, then individuals who work in the clinical laboratory, and lastly, last week was physicians, clinicians. So this week we have research development and other opportunities. And then the following week, we have some individuals coming through who are in or were in stepping stone opportunities, so roles that help to give individuals experience about that field before going on to pursue it. So thank you all to everyone who registered and provided your questions. That was really helpful. We did incorporate some of them into our session today. As a reminder, there is a Q&A box that will be at the bottom of your screen, of your Zoom screen. Please go ahead. If you have questions that come up during the session, please pop them in there and we will try to answer them during the session. You also have the ability to upvote it. So if a lot of people have the same question, I think it's a thumbs up symbol, and then we will try and to our panelists today. So before we get started, I just want to start with a quick poll. I will launch these two questions. Pretty simple. The first one is just trying to find out where everyone is at the current stage in their career. So as of September 2023, will you be a student? And then if not, what kind of where you are in your field. And then the second question is just kind of getting your familiarity about this career path, about doing clinical genomics research. So excellent. So we have about half, a little bit more than half of our individuals are not a student. We have a few that are in their undergraduate and a few that are in their PhD to high school. So a big range of people trying to have some exposure to the field, some very minor or just beginning to explore it. And the majority of the people say that they're moderately familiar, and then a few who are not familiar with this field at all. So I'll just take a minute. You all can take a look. Okay. Thank you all. Okay, so now we have our panelists. I am going to open the floor up to all of them. So in a minute, I will stop sharing my screen and we can all kind of have a informal, I call it like, it's like, this is stating myself, but maybe like the Brady Bunch screen where everyone's head is there and we can all kind of just have an informal discussion talking a little bit about their various career paths. So I'm going to have everyone introduce themselves, tell us all a little bit about who you are, where you work, kind of the education path that you pursue to get there, and then maybe one thing that you really love about your job, your career, kind of like how you got there and what motivates you. So I'm going to start in no order besides the order that I currently see people on my screen, and then we will take it from there. So Jessica, if you wouldn't mind starting everyone off.
I had a feeling it would be me because we see each other a lot, Caitlin. Okay, hi everyone. I'm Jess Giordano. I am a genetic counselor by training, so I maybe could have been on a different panel, but in my current role and the work that I'm doing now is I work as the Associate Director of Women's Genetics Research at Columbia University in the Department of OB. So I've been overall in the genetic counseling field for 15 years, and I started out in clinical practice, which was really my passion, and why I got into genetic counseling was to help patients make pivotal decisions using genetics and really impact change there. But over time, I started seeing kind of how there were still a lot of things that we didn't have available. And I started seeing research questions coming up in the clinical space. And simultaneously, a lot of sequencing was starting to become available. So in clinic, I was seeing a lot of patients, for example, that had anomalies, and all we could offer them was some basic testing, like a karyotype and then a microarray. And people just couldn't really get all the answers they needed to make decisions. So I was able to get involved in a number of different sequencing projects, both exome and genome sequencing, that have allowed us to make more diagnoses for more families and help them use that information to change their clinical management. So it's been really exciting to be in this space of thinking about, like, what are the research questions that are important to change patients' lives, and think more like translationally about research. I'm on the ground in like the operation, so like writing the protocols and the consents, managing the team of people, the IRBs, writing grants, yeah, kind of A to Z things that kind of happen in that research realm. Just by way of my own training background, I studied biology in undergrad and then went straight to genetic counseling graduate school at Sarah Lawrence. And in my role at Columbia now, I also get to work as a faculty member and teach some graduate courses. So at this point, I get to do a lot of different things, but the thing that really drives me is getting to do research that I get to see directly, change clinical management for folks. Should I tag the next person?
Sure, go for it.
Call it. I'll go to Christina.
Thanks, Jessica. Yeah, I'm Christina Pasak. I'm an assistant professor at the University of Minnesota in the neurology department. I'm research faculty, and in my laboratory, we're developing gene therapies for rare diseases. My training path, I was a molecular genetics major as an undergrad, and I loved my undergraduate research experience. I felt like that was definitely where I wanted to be, was in a laboratory. And I ended up going to graduate school at the University of Florida, where I was in a gene therapy development lab. And then I did my postdoctoral fellowship at Boston Children's Hospital, where I learned a lot about stem cells and mitochondrial function. And then when I began my own lab, I kind of combined all of these things. So we're developing gene therapies for rare diseases. We look at how the disease impacts mitochondrial function, and we use stem cells to model a lot of these rare diseases and things. And I would say the thing that I love about my career is sort of where research and science is going. It's really team science these days. And I just love that I work with so many different people at different career stages, and they bring so much different expertise to the table. I don't know any of you, these other people on this panel right now, but this is kind of a very good example of the kind of people that I work with on my teams and things like that, people who come from a lot of different backgrounds and areas of expertise. And so I feel like I'm so fortunate to get to keep learning all the time from all kinds of different people. I'll pass it on to Elizabeth.
Great, thank you. So I'm Liz Ewan, and I work at a private research foundation called Additional Ventures. And my path to this nonprofit job was I did an undergrad in genetics. I worked for a few years, went back and got my PhD in molecular genetics. Was very focused on diagnostics and working in the technology space, doing diagnostics, and that kind of merged from FISH and microarrays into NGS. And so I worked for a while at a large technology company called Agilent, and then I worked at a diagnostic company called Semaphore, specifically doing like reference lab genetics. And then now I started at this role. And I would say that one of my favorite things about being now in the nonprofit space is very similar to what I loved in all of the roles, is very similar to what Christina said, is really being able to work with a variety of different people at different stages in their career, and working on a number of different projects. I'm kind of focused on one project now, but I really get to talk to a lot of the key opinion leaders in the field, really get to understand all of the different things in terms of research and the cutting technology of like being able to administer grants and work with large data and other groups that are doing large data collection. And so really working in this very small seeming but large research community. And I will pass it off to Kumar.
Hi, everyone. Kumar. I'm currently at Novartis. Behind me, that's Patch, just in case some people are wondering. I grew up in Malaysia. I did my undergrad in Malaysia in molecular biotechnology, was faculty for 2.5 years after, then moved to Miami for my PhD for 5 years, and then did my postdoc at the Broad Institute with Arnaud Palotier, Mark Daly, and Ben Neal, who are all fantastic statistical geneticists, because I knew halfway through my PhD that as much as I love molecular biology, I think I love statistical genetics a lot better. Just looking at like how p-values can possibly tell me what signals are great. And yeah, and then after I was done, I got moved to a staff scientist position for like a few months before I moved to Novartis. Where currently I work at we actually have a reorg, but the current status is I'm still in chemical biology and therapeutics, and I'm a data scientist there working on human genetics. Basically a fancy way of saying I'm a statistical geneticist working on drug discovery. I guess I will pass it on to Alex.
All right. So I'm Alex Wagner. I have a joint appointment as a principal investigator at the Institute for Genomic Medicine, which is at the Research Institute at Nationwide Children's Hospital, as well as a tenure track assistant professor at the Ohio State University College of Medicine. I got here through an educational path through Iowa State University, where I studied computer engineering, biology, and mathematics. And then I went on to work as a clinical lab tech for a few years at the Mayo Clinic. And from there, I decided I wanted to go into bioinformatics. So I went to grad school and I got my PhD in computational genetics, studying heritable retinal dystrophies with Terry Brown and Ed Stone. Following that, I went down to the McDonald Genome Institute at Washington University, and I did my postdoc there for about 4 years. And then I was an instructor for a year as well. And there I was really focusing on cancer genomics and cancer variant interpretation. Following that, I made it here. I started my lab at IGM as an assistant professor, and I've been here for about 3 years. I primarily work in bioinformatics. I do a lot of work in a variety of consortia settings, including ClinGen, but also the Variant Interpretation for Cancer Consortium, the Hugo Genome Variation Society, the Atlas for Variant Effects Alliance, and others. And my work is really focused on how we can build computational data structures around genomic variation and all the evidence that we use to facilitate scalable genomic medicine. My lab includes a staff scientist, 3 software developers, a couple of postdocs, a couple of grad students. And we really work on data standardization. How do different groups talk about the same evidence in the same way? We do this in the consortia settings I mentioned before by doing, discussions, community feedback, modeling exercises, testing on a variety of data sets from different participants, and then ultimately turning these into computational data standards through various consortia, including the Cancer Genomics Consortium and the Global Alliance for Genomics and Health. Across all those things, I think, one of my favorite things is it's probably very much in line with what Christina mentioned earlier. Getting to work with a bunch of different people in a bunch of different settings is very gratifying. I've learned a lot about a lot of different fields in this career path. And I think the other thing that I really love about being a PI is just getting to kind of shape the vision for my own team and build up a research program that I'm very interested in and just kind of get to do the work that I'm most passionate about. I think that's all I have to say about me.
Thank you, everyone. Oh my goodness, you guys are all so amazing and all the different things that you touched. It's pretty incredible. So thank you all for sharing. A question that we often get from individuals who are interested is, I'm going to put it as twofold. So one is kind of how did you hear about your career? And it sounds like everyone kind of like started off on one path and then like took a little bit of a like lazy river ride to the different areas that they are interested in. So a little bit more about how you heard about it, and then how do you gain this exposure for someone who is just thinking about it, would like to learn a little bit more. How do you recommend gaining exposure? Or are there different conferences or resources, shadowing opportunities, etc. that you?
I'm happy to go first. So we had a Gordon conference last week that someone asked the exact question about how did everyone get onto their paths. A lot of the times well, at least if you're getting a PhD, a lot of the times the first thing you think about is, I should go to academia, and every other job is an alternative career, which I feel is not a thing anymore, at least not in the past like 5 or 10 years. That's what I felt throughout my entire PhD and postdoc. I was like, I should probably go into academia. But then I realized how much I dislike writing grants, and I dislike begging people for money for getting my own salary. Instead, what I really enjoyed was just running my analysis. So I talked to my mentors who told me, okay, well, maybe you should maybe you should be a scientist instead of a PI. I was like, that sounds like a fabulous thing. Is that throwing away all my training? And he was like, no, it's actually just using your training just in a different way that is not what you think is the default, because what you believe is the default may not be the default. It's just because it's ingrained in you that you think that's the default, but it may not be. So there are many careers out there. If you have any kind of relevancy in genetics, genetic counseling, being a scientist at any institute, being a PI, being a director, being whatever you want, as long as you follow your passion. And that's really how I fell into my job, because I liked analyzing genetic data and I followed through.
I can take the next answer. So very similarly, I went and got my PhD, thought I was going to be a PI, teach in academia. And as I was going through my PhD program, I did enjoy the research. I did get some really great advice as I was starting out, was basically to learn as many techniques and as many different types of research procedures as I could during my process. And because I had worked for 3 years prior to going and getting my PhD, I had been very focused in diagnostics and clinical genetics. So I really focused a lot of my PhD work in standard molecular biology, transcriptomics, and very different things than are used typically in a diagnostic lab setting. But my interest always was in clinical diagnostics and understanding human diseases, and considering that I really focused when I was looking whether for a PhD or for a postdoc or for a job after I graduated. I was faced with 2 really great options: a really great postdoc option and a good job opportunity. And honestly, I probably made the decision financially. So I knew that I wanted to have a family. I knew that I wanted a lot more security in terms of financial and benefits. So I ended up choosing the job role, the industry role. And one of the things that I kept doing is making sure that I worked for people that really loved what they were doing, and they could transfer that enthusiasm and really cared about not only just the product or the topic, but also about the people. So making sure that I worked with someone who cared about me as a person, who cared about not just as a person at the company or as a cog in the wheel, but just really, feeling that support really brought out the best in me. And I think that I continually followed people that I believed in, and which ultimately led me to the role where I am today, doing clinical research or creating a database of a specific disease set of patients, and following the passion and the motivation to continuously do the best that I can and give my my time and energy to something that I do believe in.
I'll jump in next. And I'm going to do that because Elizabeth said something that I think really resonated with me, which is about interest and about, you know, following your interests. And I found throughout my path, which I think was, I think this idea of kind of a lazy river to get there is a great analogy, because it's really it really is kind of, as you're going along, if you're just kind of following what it is you're interested in, I think that you're always going to find satisfaction and happiness and next steps for your career. For me, you know, I can tell a short story here. I was a graduate student. I had 3 rotations, and I remember I did my first rotation. I loved it. And I was thinking about where I wanted to go for my second rotation. I didn't really know. And, you know, I was faced with a few different labs, and some other graduate students came with me and said, oh, this researcher is, you know, really well known in their field. You should go do a rotation in his lab. You'll learn a lot. I sat down with him. I didn't really understand, you know, his ideas for, you know, what was going to be happening, but I trusted, okay, this guy knows what he's talking about. I'm going to go in there and by osmosis, I'm just going to pick this up and I'm going to do well. And it was my worst rotation. It was just awful. And, you know, and then I ultimately ended up choosing to go with that first lab that I rotated and I loved, just because I enjoyed it. And I had a very satisfying, you know, PhD journey. And towards the end of it, you know, I was thinking, oh, gosh, I guess I have to decide what I do next. And I didn't really have a clear picture in mind. I didn't think I want to be a PI. And I didn't think, you know, I want to go be like, you know, a genetic counselor. I just thought, you know, I like genome interpretation. I've been studying it for a few years. I really enjoy this. You know, and I saw this talk that Elaine Mardis gave at the National Cancer Institute about what they were doing in precision medicine. And I was very excited by it. And so I said, well, you know, like maybe that's what I'm going to want to do. So I wrote them and I ended up going to work with them for my postdoc. But you know, the path to get there, it's, you know, there are many different ways, and you know, each step of my journey has really been just based off of what I felt was the right place, the place that interested me, as Elizabeth said. And I think if you follow that, you're ultimately going to end up where you're going to be happiest.
I can follow up on that. Yeah, I really liked Caitlin's comment about, just as Alex, the lazy river, because when sometimes you hear these panels, you just think, wow, everyone knew exactly what they wanted to do, and boom, 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 it all worked out perfectly. But in the middle of it, there are many times where you're just like, oh, all right, I guess this is what I'm going to be doing next. And I think that if you know, even though people think of the academic track as being the traditional one, I actually don't know anyone who went undergrad straight into grad school, did that perfect 3-year postdoc where all their projects worked out perfectly and they got a great paper, and then boom, tenure track faculty position. I do not know anyone like that in our generation at all. So I don't think that we should think of that like traditional path for all kinds of different reasons. As Alex mentioned, there's so many different ways to get where you want to go. And I think the best advice, at least in my career, has been to just try it. And it's not a race. I mean, there are different family obligations and things. You might be limited to a certain area. You might need to make a little more money. There are various things that happen that are going to play into the decisions that you make. But I think getting into a lab and trying it, or getting into a company and trying it, because you don't know if you're going to like it or not sometimes until you get in there, and then it'll just feel right once you know.
Yes, we are unable to hear you.
Oh, sorry, I'm
Now we
using
can
the
hear you.
phone too. In that vein, I honestly had no intention of pursuing a research path at all. I mean, I did a clinical program, intended to be in the clinical space for life, and I still really love that part of my identity and my work. But I think over the years, I was just kind of open-minded to different opportunities and hearing about like how much things are changing and evolving. I mean, science is really dynamic. So I think, you know, seeing what's coming next, like listening to talks, reading papers, you know, talking to mentors and understanding like what is the next thing, and then wanting to be part of that bigger macro change, rather than simply it's not simple at all, but like being on the ground, boots on the ground is really important with patients, but also wanting to take that experience and transform that into bigger change. But I think a lot of that had to do with being open-minded, identifying mentors, like just being open to change and doing new things and not being kind of rigid about it.
Thank you. I'm going to take a little bit of a step back, and this will be a little bit of a difficult question for everyone. I'm wondering if you could tell me maybe like a typical day or a typical project. So I know that every day is probably going to be very different, but you know, some of the few things that you encounter more frequently or more common in your day work task.
I can start. My day is meetings. I mean, that's just so much of what I do as a PI. It's just talking with graduate students and staff and going on consortia calls and doing events like this. You know, like it's just a lot of communication. And then at the end of that, there's like, you know, just, you know, there's family time. Like, you know, I've got a family, I go home, I have a few hours with them. And then like when the kids are in bed, then that's when I get like real work done. I think like when I actually like do the writing and the responding to emails and all the other stuff that just, you know, has to get out the door. I just don't have time for during my normal like 9 to 5. And I have to say it hasn't always been this way. As a postdoc, I definitely had more time for that during like regular working hours. As a graduate student, I had way more time for that during regular working hours. But I feel like at each career stage, like your available time to do anything other than, you know, talk to people about their projects, but what they're doing, about how you can collaborate, those sorts of things. Like any other time, do anything besides that kind of goes away. And so one of the key strategies, I think, to, you know, being a successful in this field is figuring out how to protect your time a little, which I'm failing at. But, you know, like I'm sure there are people out there that have done this successfully.
I'll go next.
That was,
or Jess, you can go.
I just want to say that was very, well, I was just going to say that's very relatable and honest, but you can go ahead, Elizabeth.
Yeah, so my research project is just kind of getting off the ground. So we're doing a lot of logistical planning. So one of the overarching goals is that we're going to collect 5,000 patients and their family members to do whole genome sequencing on them for this specific disease. So we have a lot of moving parts, a lot of vendors, we have deadlines, we have other internal company goals, so grant cycles. So it's a lot of logistics at the moment. I think a lot of meetings, a lot of communication, a lot of planning and future proofing as much as possible. But I have a feeling that once our project launches, it will be kind of what I'm used to, like fire putting out and logistical planning of, all right, this data didn't move, it doesn't look the right way, who's accessing it and how, and what are all of those kind of things. So a lot of planning at this point. I will say that one of the consistencies like for all of the jobs that I've had is, again, the communication, and then the liaising between maybe the labs and the people doing the sequencing, the bioinformaticians and the statisticians, and really trying to understand all of the different aspects as a kind of central person to making that connection.
I could go. So in line with the idea of meetings, I guess to add color to those, they're really about, I have a lot of meetings as well, but it's frequently about such a range of different topics, like from the top of a project like conceptualization, brainstorming with people, you know, then working out, writing a grant. And this could all happen in one day because you have a lot of different things going on. You might also have like a mentorship meeting with somebody. You might have a team meeting and think about all the operational logistical challenges. You might have to spend some time working, you know, on a response to the IRB, for example. Today, for instance, I'm also going to be teaching. So there's just like a lot of different things, but I do think underlying a lot of it is communication, really, like Alex pointed out. But I think it's really fun that like you can wear a lot of different hats within one day, which is really exciting, and you get to meet a lot of different people.
I have nothing to add. Alex described my life perfectly.
I wish I had something else to add other than I'm in meetings all day.
Yeah, meetings can take up everyone's schedule. I know you've all talked about this love of your career, which includes constantly learning, constantly interacting with other people and their different stages of their field. So can you tell us a little bit about how you all stay up to date with the current, you know, information and the changing technology and the future of genomics? What are some of the resources that you use, or maybe colleagues that you lean on, or kind of tools that you have adapted to continue to stay up to date with everything?
I'm happy to take a whack at it. I always lean on to my previous colleagues at the Broad because they're usually at the forefront of these things. So like Slack messages, one of my favorite ways to get in touch with things, because as much as I want to read every paper there is out there, is going to conferences. I don't get to go to as many conferences being in a pharmaceutical company as I used to being in academia, but at the same time, I try to make the biggest bang for my buck, if you will, every time I go to a conference and absorb as much as I can during the conference and outside. And whenever I get updates from like colleagues, I usually just like forward it over to other colleagues at Novartis. Another good resource that I keep in track with, especially in statistical genetics, is Twitter. And yes, we've tried to like not use Twitter over the past year, but we've tried and failed at Mastodon, and some of us have tried on the new thing on Instagram called Threads. Yeah, even like yesterday, we forwarded something on Twitter by Viraj Rajagopal from Regeneron over to some colleagues of ours about some really cool signals in FTO. And I thought that was really cool because FTO is like known as the obesity gene. This is one variant there that has been constantly associated with obesity. However, when shown in this paper, when you do a knockout in mouse or in mice, the mice show a very discordant phenotype. So I thought that was super fascinating. Nothing to do with what I'm working with, but just like, oh, kind of cool. Anyway, those are my resources.
Yeah, I can go next. I think that part of why we build these teams of diverse people and backgrounds around us is because we can't alone keep up on this stuff. The fields are just exploding. And so I use things like Slack, like Kumar mentioned, and things, and we put articles in there and blips on, you know, what did I like about this? Why is it relevant to project A or B or whatever it is? And a couple of people through our discussions have mentioned mentors. And one thing that I think it took me a while to understand is that having a mentor isn't a mentor, and it's not this one official person that you're assigned. Over time, you end up with mentors for different reasons. Like certain people are going to be really good at advising you on maybe work-life balance, and they're going to help you with that. And then other people are going to advise you on negotiating, and then other people and not any one of these things is going to come from the same person. And then I think in the survey in the beginning, a lot of you might be undergrads or your various early career stages. I would say also don't be shy about coming up to anyone like us, because students are like the best. You're the ones we really want to talk to and encourage and things like that. So never hesitate to approach someone who you think their career, you find it intriguing or you want to know more. That's like always welcome.
Yeah, I was gonna say, like, just not being afraid to ask questions. I think that's how I learn the most, because I agree, you can't read every paper. You can get a digest and read the headline sometimes, and that at least pushes you in some direction to keeping up to date. But not just going to conferences, but really like talking to people and asking questions. And don't be afraid and think it's a silly question or not a relevant question. Like just asking questions, and even like Christina said, like the team around you, asking them questions. Like the folks that I work with that do a lot of curation, they might be more up to date on the newest tool that I haven't seen in a while. So really just asking around and being curious is important.
Yep. I would say I have a number of things that I follow. I follow certain people on LinkedIn. I read Genome Web pretty regularly just for summary, blurb, snapshots. Also Twitter. My colleagues and I share papers. Those are usually very disease and work focused. But I will say that I've really prioritized keeping in touch with as many of my friends and colleagues that I have met throughout my process, because even though we don't necessarily work on the same things, we all work in different places, whether it's drug discovery or education and academia. Like really, I cherish all of those conversations, and I really don't think that I would have thought of a lot of the things that I think about when I hear about my colleagues that I went to grad school's jobs and those connections. And then always like, I keep that door open, because when I do have students that come talk to me, I then have that connection that I was like, oh, I know someone at Novartis. I know someone at Biogen. I can connect you with them who work on zebrafish or this, and really making sure that I put those relationships as a priority as well. And that's actually how I got the job that I got today. I had been laid off from Semaphore, and I was just kind of blindly applying to any and every job that I could think that I was qualified for. And someone else had interviewed for this job that I'm in currently, and they were like, well, this sounds like a really great job. I'm not interested, but I know someone who would be. And if it wasn't for that connection, I probably wouldn't have even, this job wouldn't have even come across my desk. So making sure that not only am I keeping other people in mind, but that I'm in other people's minds as well, is one of the things that I would recommend as a very big priority, where it's a small world, small fields relatively. And most people that I've met have all wanted to help and be helpful.
I think most of the primary ways by which I stay up to date with info in my field have already been mentioned, right? Working in consortia settings, I think was where Christina was going. And going to conferences, as Kumar said, and networking, like Elizabeth said, like these are all like the primary means by which I stay up to date. The only one I didn't hear, which I'll just add in, is taking advantage of opportunities to review papers. So that is a great way to see not only work that's usually pretty targeted towards your area anyways, by virtue of you having the opportunity, but also in the process of reviewing it, you'll often see references to other related works, because those authors just did the background reading for their own paper, and it's a great way to see like what's happening right now. So if you get these opportunities, it's really not just an opportunity to help advance the field as a independent reviewer, but also a means by which you can stay current. So it's a really nice opportunity for those of you that are in a research oriented setting to stay up to date.
Excellent. Thank you. Those are really great resources for us all to know and use and continue to remind ourselves about. So taking a little bit of other question or another approach, tell us a little bit about what you find most challenging or the most challenging part of your career or challenges that you had to overcome in order to kind of get to where you are at.
It's the same answer. It's meetings. But no, I think time management is it really is. It's a big hurdle. There's a lot that's asked of you, I think, in the research field, and finding ways to balance everything, I think is really key. And that's probably true for lots and lots of disciplines, but it's certainly true in this one.
Yeah, I would agree with that. And I think that something that's interesting is that our projects never end. My brother is an engineer, and he's like, well, we did that project, it's over, and now I'm doing this project. And he just doesn't understand how I've been on the same projects for so long. And it's just, it keeps going and growing, you learn more. And I love that about this career, but also it never ends, which makes the time management part hard, because at the end of the day, there's still a long to-do list, and that's how it is.
There are some operational things that I just really don't like, and I'm glad there's people in the world that do. So things like material transfer agreements and data use agreements, there's a lot of kind of legal things that go into doing research, especially multi-center collaborative projects. And I'm really happy that I have people that like doing that, but I don't like doing that. And sometimes I have to, and they can be a little cumbersome to get done. So some of that is really not my favorite, and just the reality. But other people can do it for you. So.
I guess one of the things I'll add is maybe it has to do with the logistical and legal type of things. Initially, we embarked on this very ambitious project at the Research Foundation, and it seemed pretty straightforward. We're going to sequence 5,000 people. We're going to make the data publicly available. It's going to be great. We're going to have the largest data set of this kind. And then when you actually get down to the details, there have already been, some of these people sequenced. How are we going to identify them? What data is going to be available? What kind of clinical data are we actually going to be able to get? How are we going to be able to make it useful? How will it align with all of the other studies that have preceded ours, so that in the future, we're not just adding to the noise of genomes that live in this world, but can actually be ingested and manipulated and accessed for what we actually want it to be used for. So there's a lot of the really what seems like minutia details that are very big roadblocks to actually succeeding in the overarching projects.
So I was not clear if the part of the challenges was leading up to the career or during the career.
Can be both.
Okay, well, leading up to the career, it was a lot of rejection. Like that's one of the things you go through so much that nobody really talks about. Like going back to what like Caitlin, Alex, and Christina said, it's like that lazy river, right? That lazy river has a lot of stops in between. Like when I was trying to get a PhD position, the number of schools that said no to me because I came from a 3-year college instead of a 4-year college was insane, because America doesn't like 3-year colleges. Go figure. And then like, going through your grad school experiments failing, going through postdocs where papers get rejected or grants get rejected. And then working at Novartis, like, maybe hearkening on what Jessica said of just like the organizational stuff. that's the stuff that I don't want to be bogged down by, which you think that me wanting to focus on more science, I'm still getting bogged down by. Like I moved away from that kind of career to just focus on science. But that's really what I dislike the most, or having like probably a stop in my job on a daily basis. Like just like I need to get all these other things done, but the science waits because I have to get all these things done, right? Or waiting for things because they have a long approval time. And when you follow up on people and they're like in a different time zone, or because they live in Europe and they have 2 months that they take off in a year, and you have to wait till the end of August, and then you get that thing reviewed, not even approved. So, you know, there are things that you kind of like figure out along the way. But yes, so those are the frustrating parts about my job, at least, and leading up to my job.
I think projection is hard, no matter who you are, what field you are in. So thank you for opening it up, bringing it, kind of normalizing it, and saying like, it happens to everyone, and it will be okay. And you all here talking to us today are a real big testament to it. So I appreciate that very much. So my final question that I'm going to ask for everyone is to give us a little bit of your idea of the future of your field, where you think this is going in, 5-10 years. I know we've gotten a lot of questions about how do we think that artificial intelligence may impact kind of what we do or where we work. And so I'm sure if anyone has any thoughts, or could be AI related, could be totally unrelated to where you think the future, or what students approaching this field should maybe think about, might become more in their like scope of practice.
I'm going to ask a clarifying question. When you say this field, are you talking about like our individual fields of study, or are you talking more about like the scope and mission of ClinGen?
Yeah, so I would have you focus on your individual career.
Okay, well, I'll start. I like you brought up artificial intelligence, because a big part of what we're focusing on in the work that I do is data structures and data semantics. What does a given value mean? You say breast cancer, what do you mean by that? It's actually quite fuzzy when you start like boiling it, like, tearing it down. And there's a lot of complexity about how different systems organize data in different ways. And this is one of the challenges when you're trying to build machine learning models, build AI on top of them, to talk about concepts in genomic medicine. It's that, a lot of that noise can make it very difficult to actually do learning on the data. And so, one of the things that I think that the NIH has been focusing on is how do we do a better job of constructing data sets so that we can do machine learning and AI on them. And they've got a couple of big programs. One of them is the Bridge to AI program, which is focused on exactly this question. And I think it launched a year or so ago, and it's part of, I think, a larger initiative in terms of our national interests, or our national funding interest, to try and address this challenge of AI ready data generation. Earlier there's a comment that Elizabeth made that I think also kind of points to this, which is, we've got our data set, and we're thinking about how this fits in line with all the preceding studies, and and how can we make this better for people down the road, right, that want to do the same thing. And this is really what that's getting at. How do we structure the data in a way that makes it easy to then pick that data up and use it in larger meta-analyses and machine learning and AI? And I think the future of our discipline is really advancing our practices and validation for data sets that are generated in order to enable those types of machine learning driven outcomes.
I can go next. I think this is an excellent question. It's one that in my field, I'm asking myself all the time. I'm trying to anticipate where things are going. And one way in that which this comes up with for the teams that I'm working as part of is we're trying to translate gene therapies. So we're thinking about what the FDA is going to want to ask us when we are asking for investigational new drugs and trying to get through these meetings and things. And it's constantly changing based on what happened with the most recent clinical trial for XYZ. And so you're always trying to stay on top of what's going to be expected, even though I have to sort of anticipate what are they going to ask me for 2 years from now. And the answer is really, I don't know, but I can make, an intelligent guess and try to be as prepared as possible for what's going to come up. So I guess it's not really an answer to the question, but it is a question that I'm asking all the time and thinking about very often.
I could go next. I'm in the women's genetics space. And so I think the two big things that I see coming and the work that we're doing is really focused on it, is moving away from diagnostic procedures, CBS and amniocentesis, but taking all our learnings about, curation and interpretation, and then being able to do genome level interpretation non-invasively. So allowing individuals who are pregnant to get access to the fetal genomic information early in pregnancy without doing a procedure. I think that's exactly where we're trying to go, and I think it'll happen in our careers. And then for people that are not going to do any prenatal diagnosis, I think we are going to be seeing, and there's already large studies doing this, genome sequencing as a newborn screen. So, moving away from kind of the tandem mass spec and some like simple assays, but really thinking a more genome-wide approach to make sure kids, newborns are getting diagnoses early and gaining access to interventions and treatments that can really improve their outcomes.
Yeah, I like what you said about also access. I think one of the reasons I even shifted in careers was because I think a lot of the insurance companies are trying to figure out how they are reimbursing all of these and what tests are the most valuable and pay off the most in the future. So, companies that offer diagnostic companies that offer a really wide variety of tests really are kind of going by the wayside, and having a single test that can then be sliced and diced for specific needs is kind of where this is going, but we're kind of lagging behind because those tests are very expensive to do and that reimbursement level is not there. So understanding for me, what is the diagnostic landscape look like in terms of genetics, and how can the data and the research really support the defense of these critically important tests for our population to keep the needle moving forward.
So happy to go next since there's no one else. So piggybacking on what Alex and Elizabeth said, so like from the statistical genetics point of view, a lot of us are thinking about like what AI can empower us to try and figure out new drug targets. Because that's really what my job is at the end of the day. Find a drug target, talk to the bench scientists, see how could we validate it. Because we have so much data. It's just a matter of trying to figure out where in the data that we could pinpoint, and could it make a good drug target? Could we also repurpose a different drug, right? And this has been really where I think the field has been moving for the past few years. But something else that Elizabeth brought up is just how do we make this more accessible to everyone? Because right now we know that most genetic data is made up of white Caucasians, and we know that that's not the population diversity or breakdown globally. There are 2 papers, one just recently came out with an update from this. And I think about this a lot, about how at Novartis especially, we could try to improve our diversity efforts during our drug discovery projects, during our preclinical estimations that we do at the end. And we are moving forward. It's just, there's only so much. Like, even though we have so much data, we still need more data to try and figure out how is it we can target drugs to people who are not being helped right now. There was a paper that just came out this morning from UCLA where they looked at the different diverse populations just in LA by itself. We know that LA has a high Hispanic population, but within the Hispanic population, there's high admixture. Are we targeting them well enough? Because we realize there are some allele specific associations to diseases that would otherwise not even be observed in any other population types. So those are the two things that I think are possibly going to make a difference in the future. But yeah, those are my 2 cents.
Thank you. Thank you all. This was a lovely discussion. So a huge thank you to our panelists. Thank you, Kumar, Christina, Jessica, Alex, and Liz. Thank you for your time and sharing your experience with our audience. Thank you to our attendees for joining and for sending in your questions. As a reminder, all of these recordings, all these sessions are recorded and posted to our website and available for people if you want to re-listen or if you are unable to attend live. So next week we have our last session, and that is for stepping stones opportunities. All sessions, so next week, same time, 12 p.m. Eastern on Wednesday. Let me put in the chat, this is the link in case you want to go ahead and view any of the recordings that you were unable to attend. There are also the recordings from years previously. So those are all excellent resources for you guys. And lastly, at the end, so after next week, we will be sending out a survey to all of our registered attendees. I really appreciate your feedback, letting us know what was good, what needs to be changed. We try and do this career series every year, so your feedback is taken into consideration, and we like to modify it moving forward so that we can make this the best opportunity for any interested learners. So again, a huge thank you to our panelists. I really appreciate your time. Thank you for sharing your expertise, thank you for opening up about your career path, the good things and the bad things, and what you are expecting for the future. So let me know if you have any questions. And again, I really appreciate everyone's
Thank you.
Thank you.
Thank you.
time. Thank you.
Thanks, all.